So with this, without further ado, let's get started with Henrike Niederholtmeyer. She's an Eminöta research group leader at the MPI for terrestrial microbiology in Marburg, and she will talk about engineering protein dynamics in artificial cells and organelles. So Henrike, if you can, exactly.
Thank you so much, Philip. I will share my screen. All right. Can you share? Can you see my screen?
Yes, it looks great.
Okay, perfect. So my talk is probably going to be a little bit different from most talks in this conference in that we are not looking at natural dynamic systems, but we are building or aiming to build synthetic ones. And I'm going to give you some examples on how we are engineering protein dynamics in artificial cells and organelles. So I'm interested in cell-free synthetic biology, and the goal of this is to simplify the complexity that we have in natural living cells. Let me just get a laser pointer, and take isolated components from biological systems and rebuild simplified systems in test tubes or other cell-free systems. And the goal of this is to have a more simplified and controllable reaction environment where we can more easily make predictions and engineer more easily. And another goal of this is to eventually build enough complexity to arrive at something that resembles a living cell. If you compare a cell-free reaction with a living cell, of course, a striking difference is that in a test tube, you have basically a soup of molecules where there's no spatial organization and no temporal control over when reactions happen. And what I'm interested in is to rebuild mechanisms for spatial and temporal organization in these test tube systems. The tools we're using is cell-free transcription and translation reagents. We build synthetic gene networks in these reagents, and we're also using synthetic materials to assemble containers that resemble cells and organelles to spatially organize reactions. And for all of these experiments, we're using different microfluidic tools to control the reactions. At first, I'll give you a short introduction into cell-free transcription and translation reactions. You're probably familiar with them, but in case you're not, the idea is that you have all the components that are necessary to go from a DNA template to mRNA and finally a protein combined in a test tube with the necessary precursors. If you're working with E. coli-based cell-free transcription and translation reagents, you have the choice between either a minimal defined reaction system that's commercially available as the pure system. It contains the almost 30 proteins that are necessary for translation, purified from E. coli. And of course, the system is great because you know exactly what's inside and in what concentration. The other choice is to use lysate-based systems that are basically just ground up E. coli cells. Of course, these systems are a bit more of a black box, but the advantage is that they tend to have higher expression rates. And in these cell-free transcription and translation reactions, you can not only express one protein, but you can actually use them to assemble gene networks. And with that, I mean that the product of one gene regulates the expression of a downstream gene. We were really inspired by the early examples of synthetic biology. For example, the repressilator. It's a very simple network where 3 repressors repress each other in kind of a ring architecture. And that leads to oscillations of gene expression in E. coli. And we were wondering if we can also run these dynamic gene networks in cell-free transcription and translation reactions. And it turned out that it's difficult, especially in test tubes, because these TXTL reactions, they go to equilibrium very, very quickly. So after 3 hours or so, you basically have no synthesis activity at all anymore. And that is because you run out of precursors, and especially because reaction products accumulate. So we reasoned that we need some kind of reactor where we have a flow of fresh reagents, and those fresh reagents replace part of the old reaction volume. Like that, we reasoned we could keep transcription and translation activities at steady state levels for extended periods of time. And because cell-free transcription and translation reagents are laborious to make and expensive, we designed a microfluidic device that fulfills this role as a flow reactor. Here you can see the device in action. So we have PDMS valves that precisely control how much fresh reagents we're adding into the reactor, that is this ring. It just has a volume of 30 nanoliters, and then pump the reagents in a circle to mix them. And in this device, we can actually assemble dynamic gene networks. So here we designed an oscillating gene networks where we have a positive feedback loop that's coupled to delayed negative feedback. And you can see that this network was oscillating nicely for more than 30 hours in the device. And also our initial inspiration, the repressilator, turned out to be working when we combine it with a lysate-based TXTL system. Here we're looking at experimental results where we're looking at the expression activity of the 3 different promoters in the network. And also this one was oscillating nicely for extended periods of time. So we decided to expand on the repressilator network and to build larger networks built from more repressors. And theory predicted that even nodes of repressors arranged in a ring should lead to stable steady states, and odd numbers of repressors in the network should lead to oscillations. So networks like 3 nodes, like the repressilator, or 5 nodes should lead to oscillations. And that's exactly what we saw. So in the network built from 4 nodes, we could use 2 different inducers to switch the system into either of the 2 stable steady states, as you can see here in these 2 graphs. And then the networks we built from 5 nodes, they were oscillating, but with much longer periods than the 3-node repressilator network. So I showed you how we can run dynamic gene networks using cell-free transcription and translation reactions and microfluidic reactors. And now I'm going to tell you a little bit more about our efforts on using synthetic containers that mimic living cells or organelles to organize reactions in space. And here's one example of how we're programming these reaction containers with synthetic gene networks. The goal is to eventually use complex dynamic gene networks like I showed you in the first part. But for that, we still have some engineering to do. And here's a simple gene network that we've already demonstrated that's working. So we could use communication between the cell mimics where there was a transcriptional activator that led to activation of gene expression in neighboring cell mimics. We produce these cell mimics in a microfluidic device where we combine, in a microfluidic junction, we combine flows of an inner aqueous solution with an organic phase and another aqueous solution to form double emulsion droplets. These double emulsion droplets encapsulate DNA that we later use to program the expression in the cell mimics, and they have an oily organic shell that we polymerize to form porous membranes. We collect these droplets from the chip and treat them with UV light, and that induces polymerization of the acrylate monomers that we have in this shell. We then treat them with a mixture of buffer and ethanol, and that permeabilizes the membrane and also induces the formation of a hydrogel structure in the center of the cell mimics where DNA is immobilized. So this is how the cell mimics look like. When they are produced, they have approximately the size of a eukaryotic cell. And they contain an artificial hydrogel structure that we call nucleus because it contains the genome of these cell mimics. It's composed of a synthetic clay. Here you can see how this clay looks like in bulk. And these clay minerals are very efficient at capturing DNA from solution and holding on to it tightly so that it's immobilized in the center of these cell mimics. But at the same time, the DNA is still accessible for transcription. And so when we add transcription and translation reagents, we can express genes that are that we encapsulate in the cell mimics. The cell mimics have very porous polymer membranes. Here you can see some SEM pictures of the membranes. And because they are so porous and permeable, we have an exchange of molecules between neighboring cell mimics. And so we can program communication or interactions between the cell mimics. Because the membranes are so porous, it also makes it easy to start transcription and translation. We can simply add the reagents on the outside, and then they diffuse inside, and genes that are encapsulated in the cell mimics get expressed. Here you can see how we were expressing a fluorescent protein. That was encoded in the DNA in the hydrogel nucleus. This reporter was also fused to a DNA binding protein, and we are providing many binding sites for this DNA binding protein on the DNA so that the protein eventually gets localized in the cell mimics. And that's why you see these artificial nuclei light up in green over time. Because the membranes are permeable, our cell mimics are kind of coupled to each other, and mRNA and protein can diffuse from the cell mimic where it's produced to neighboring cell mimics. Like that, they can communicate with each other with transcription factors, and those can then act on the transcription in neighboring cell mimics. And when we arrange these cell mimics in large, dense communities, we can see how protein gradients form. So in this case, we were using sender cell mimics that were producing a protein, and then you can see it spreading out into the surrounding receiver cell mimics. What we're interested in now is to use the more complex dynamic gene networks that I showed you in the first part of the talk, to use these gene networks like oscillators to program expression in large communities of cell mimics. And I want to give you one last example of how we are programming protein dynamics in cell-free reactions using synthetic organelles. we found that a system composed of lipid sponge droplets can act as a very programmable self-assembled spatial organization in cell-free reactions. And this was done in collaboration with Ahanjit Bhattacharya at UCSD. So we found that when we mix in solution a synthetic glycolipid that has a galactose head group and a single oleic acid chain, when we mix it in solution with non-ionic detergents like IgPel or Triton X, then we get a turbid dispersion. And when we look at it under the microscope, we see these microscopic droplets that have a high optical density. And these droplets are also very dynamic. Very often we can see them fusing, and after a while they coalesce into a single liquid phase at the bottom of the tube. These droplets are not vesicles. They are composed of a lipid sponge phase. So vesicles have a lamellar membrane, so they are closed structures. And our sponge phase droplets, in comparison to that, they contain a sponge phase where you have a very dense membrane network that's contained in the droplets throughout. So when we use a lipid dye, the droplets are stained throughout because the membrane network, because the droplets are composed of this membrane network. But at the same time, they also contain high water content, and so we can also target soluble molecules into these droplets. And like I mentioned, they are very dynamic, and we can often see them fusing under the microscope. These droplets are very biocompatible, and they can take up proteins, like I mentioned, and I'll show you some more examples of that. And here, this is just an illustration of their biocompatibility. We can add them to cell-free transcription and translation reagents, and still protein expression works very well. And we can also express proteins that have an affinity to the droplet phase. So in this experiment, we were expressing a lectin from Pseudomonas that has an affinity to galactose, and galactose is on the head group of the synthetic glycolipid. So the protein, when we are expressing a fusion to GFP, gets sequestered into the droplets during its synthesis. Here's a control. If we are just expressing GFP alone, it's excluded from the droplet. So it's the specific interaction of the lectin with the glycolipid that recruits the protein into the interior of the droplets. Most proteins don't have an affinity for the droplet phase. And so we had to find a way to target specific proteins into the droplets to program co-localization of different proteins and reactions into these droplets. And the way we can do this is by adding, by doping in small amounts of lipids that have an affinity handle. So in this case, we're adding a lipid that has a nickel anti-A group, and then we can target his-tagged proteins into the droplets. So here, for example, we were adding GFP with a his tag, and that then partitions into the droplets with a very high efficiency. And like this, we can co-localize proteins of our choice into the droplets and program reactions inside. We can use the same strategy to target other molecules, like using a biotinylated lipid, streptavidin, or through sandwiching with streptavidin, other biotinylated molecules such as DNA. I see that I'm almost out of time, but this is the last example. So in cells, proteins often dynamically shuttle between different cellular compartments, like, for example, the nucleus and the cytoplasm, and these localization changes are often very rapid and reversible. And we wanted to see if we can program something like that in our artificial organelles. So we encapsulate a photoreceptor from Arabidopsis, phytochrome B. On this protein with light, we can switch it between a non-binding and a binding conformation. In the binding conformation, it interacts with phytochrome interacting factors, and we can use this as a tag to recruit proteins like GFP into the droplets. Here in this movie, we were switching between far red light and red light. And you can see that GFP tagged with a PIF tag gets encapsulated and then really rapidly released from the droplets. And these changes are reversible. Here you can see a change. And this changes the concentration of GFP in the droplets. The changes are very rapid and reversible. And also in solution, we can control the concentration of GFP that's available. And this we can use to control reaction rates of biochemical reactions in the solution. So we were reasoning that we should be able to use this concentration change to control a reaction. We were using ClipXP, that's a very large protein complex, and it's so large that it doesn't fit into the pores of the sponge droplets. So degradation can only take place outside of the droplets. And then GFP should be protected from degradation when it's encapsulated, and available for degradation only when it's released from droplets. And that's what we can see when in red light, the degradation is much slower than in far red light, and we can also switch the rate of the degradation by switching the wavelength. Yeah, so I've showed you how we can use gene networks and cell and organelle mimics to control protein dynamics. And most of this work was done in the Devaraj lab at UCSD. And now I'm setting up my own lab at the Max Planck Institute in Marburg. And feel free to contact me if you have any questions later on. And if there's time, I'm also happy to answer some now.
Thank you very much, Henrike, for this great talk. Yeah, so to everybody, also to the other presenters, of course, if you have any questions, please either raise your hand using the raise hand tool, or post the question in using the Q&A button, and I will ask them for you. Maybe I want to start with a small question myself. So initially you had this this cute picture of, adding new energy to the mix by replacing some of the reaction volume. But somehow there's a balance between adding enough energy, but not taking away too many products in order to disrupt the regulatory mechanisms. So can you say anything about this trade-off and how the different experimental realizations you presented deal with this?
So it's actually very good to replace some of the protein products as well, because that allows you to actually get dynamics. And we are kind of setting the dilution rates to comparable rates that you would get from dilution by cell growth. So we can set dilution rates that match the dilution rates in E. coli by cell division. And for example, with that, we can tune the period of oscillations. The faster we go, the faster the oscillations.
Okay, thank you. So currently I can't see any other questions, which due to my short introduction actually is fine time-wise, but if people who joined later, because some people joined a few minutes after the start, if you if this was too quick to post your question, I said initially, all of the speakers are also happy to be contacted by email if you have a question. And otherwise, maybe you can also look in one of the lobbies for them after the talk. Actually, yeah. So with this, I would say thank you again to Henrike for this great talk. Thank you.